So in this lab, we are going to look at the diversity of life. And just to give you an overview of what we will be doing in this lab and what we want you to gain from this experience, the objective of the lab is that the purpose is to explore the diversity of organisms on Earth. The method that you'll use to do this objective is that students will observe photographs of related organisms to identify the organism's taxonomic name based on their shared characteristic using a dichotomous key. And I will go into more detail about this in a moment. The outcome, what you should walk away with after this lab, after completion of the lab, students should be able to identify which taxonomic group an organism belongs to based on its shared characteristics. So basically, we want you to have a general understanding of how organisms are grouped and what shared characteristics they share. So what we're looking at in this diagram is a broad overview of how classification works. And so what we're looking at is that life can be divided into 3 domains. And so if you look at the bottom of this diagram, you'll see what we call a universal ancestor. So this would be like the first living cell. And so that first living cell branched off into multiple domains. The first domain is domain bacteria. Bacteria is probably one that you've heard of many times. We have what are called gram-positive bacteria. That would include things like Streptococcus pyogenes. So if you've ever been, sick with strep throat, well, that's because you have an infection of a gram-positive bacteria. But when we think of bacteria, oftentimes we have this negative connotation, meaning that we think of bacteria and we think of disease. But what you want to be aware of is that most bacteria actually don't cause disease. And in fact, they have there are a lot of useful uses for bacteria. If we look down here at the bottom, there's one called cyanobacteria. Cyanobacteria is an aquatic bacteria. It is able to do photosynthesis. And what that means is that it's able to make its own food. It takes carbon dioxide and water and sunlight, and it's able to produce food, meaning glucose. And a byproduct of that process is going to be oxygen. And so when you think of the oxygen in the atmosphere, probably the first thing that comes to mind is plants. But in fact, cyanobacteria are a main contributor to oxygen in the atmosphere. And so they play a very useful role. And in fact, in your body, you have lots of different bacteria that are part of what you call your normal flora or your normal microbiota. And in fact, you couldn't survive without that bacteria. There have been many diseases that have been associated with disruptions to your normal flora. Anything from obesity to autism to diabetes, the list goes on and on. There are a lot of diseases that are associated with problems or shifts in your normal flora. Next, we come to domain Archaea. Domain Archaea, these typically are what we call extremophiles. Not always, but usually. And what that means, if we say that they're an extremophile, that means they are an extreme lover. They live in environments that most other organisms can't tolerate. So we have our methane producers, we have salt lovers, so types of archaea that can grow on salt crystals, for example. We have our hot acid lovers. These are they're able to tolerate extreme environments. Now, when we look at domain bacteria and archaea, one of the things that they have in common is that they both are prokaryotic cells. And what you're going to see, what that means is that those are both organisms that are unicellular. They exist as one single cell, and that cell lacks a membrane-bound nucleus. That's what makes them prokaryotic. The next domain is domain Eukarya. Domain Eukarya, like the name suggests, these are our eukaryotic cells. These are cells that do have a membrane-bound nucleus. The DNA is contained within a separate membrane, and that is different from domain bacteria and domain Archaea. If we look within domain Eukarya, we can divide it into the next level of classification, which is called kingdoms. Now, historically, there were 4 kingdoms. That was one of the historical types of classification. And the 4 kingdoms that you often hear are Kingdom Protista, Kingdom Plantae, Kingdom Animaliae, and Kingdom Fungi. Now, the reason I say that that's the way it used to be done is that Kingdom Protista has now been broken out into several other kingdoms. And the reason for that is that the way that an organism was classified as being a protist was if it was a eukaryotic cell, but it wasn't a plant, it wasn't an animal, and it wasn't a fungus, it was put in this other group that they called Protista. Think of it like if you have a house or if you have a drawer in your house, that is your junk drawer. So I have a drawer that when I'm cleaning up quickly and I don't know where something goes or where to put it, I just stick it in the junk drawer and I figure I'll deal with it later. That's what Kingdom Protista is like. It's the junk drawer. Scientists didn't know what to do with these organisms. They were eukaryotic, but they weren't a plant, animal, or fungus, so they put them in this other group. They put them in Kingdom Protista. But now scientists are starting to go through and separate Kingdom Protista into separate kingdoms. Because in fact, the way that they were classified as being Protista, there weren't a lot of similarities between the organisms. So it wasn't the best classification scheme. Within Kingdom Protista, we have amoeba, we have flagellates, we have dinoflagellates, which can cause a red tide. We have diatoms, we have algae is also in Kingdom Protista, etc. Kingdom Plantae, we're going to learn a lot about Kingdom Plantae. Kingdom Plantae are plants, right? If you think about plants, you want to think about what plants have in common. Well, they are able to do photosynthesis. They're able to make their own food. And we'll go through and talk more about what they have in common in a little bit. But some examples of plants, we have our flowering plants, so roses, lilies, any of those flowering plants that come to mind. We have our evergreens or our cone-bearing plants, like a pine tree, for example. We have ferns, we have mosses. Those would all fall under Kingdom Plantae. Kingdom Animalia, animals. You can probably list an infinite number of animals. We have mice, lizard, fish, sea star, snail, humans, jellyfish, right? The list goes on and on. So there are lots of examples that you can probably think of for Kingdom Animalia. Kingdom Fungi, these are your molds, mushrooms, and yeast. If it's a mold, mushroom, or yeast, it's going to go in Kingdom Fungi. These are organisms that are what we call heterotrophs. They're other eaters. And in fact, many of them are what we call saprobes, which means they feed on dead, decaying matter. And again, in a little bit, we'll talk about what those groups of organisms have in common. So when we talk about our classification schemes, the easiest way to think about the way that we classify organisms is I remember a sentence to help me remember the order. And that is do, which is domain. And in this case, this would be Eukarya. Do, keep, pots, clean, or family gets sick. So this is how I remember the classification scheme. So do, keep, pots, clean, or family gets sick. So do is domain. Again, the most broad type of classification. So in domain Eukarya, for example, you would have plants, animals, fungi, protists, etc., right? It's a very broad classification. Then we get to kingdom. Now, kingdom is starting to get more specific. So in this kingdom, we're looking at kingdom Animalia, animals. So if you look, we have our worm, we have our fish, we have our bear, we have cats, we have lions, we have butterflies, we have mice, right? These are all types of animals. So now we've excluded the plants, we've excluded the fungi, we've excluded the protists. So we're starting to get more specific in our classification. After kingdom, we come to phylum. And phylum, in this case, is going to be Chordata. These are organisms that have what's called a notochord. A notochord is a precursor to a vertebrae. So in this case, now the worm is out and the butterfly is out because they are not precursors to or having a vertebrae. So those are out. So we still have our fish, we still have our bear, we still have our cat, we have our lion, and we have our mouse, but we've already excluded some. Then we get to class. Class, Mammalia, mammals. These are grouped by the fact that females have mammary glands to feed their young, right? If you think of humans, we fall under the class Mammalia. We're mammals. We have mammary glands and we can feed our young with our breasts. So now the fish is out, okay, because the fish is not a mammal. The other thing about mammals, hair. So bear is still in, cats are still in, lion is still in, and the mouse is still in. So, but we've gotten more specific. We get to order. Order in this case, carnivora. Carnivores. They eat other animals. So now the mouse is out. We still have the bear. We still have the cats. We still have the lion. Next, we get to family. In this case, our family is Felidae, felines. So it's the cat group. So the bear's out. Then we get to genus. And the genus in this case is Felis. So now these are more of kind of, more house type cats. So lion is out. And then we get to species. And species is the most specific. So think of species specific. So in this case, the species Felis domestica. Notice that the name is what we call a binomial. Bi means two, nomial's name. Binomial nomenclature. So when we talk about organisms, we give them a binomial name. So this cat would be in the species Felis domestica. Now, I'm going to give you a biological definition of what is a species, right? How do we classify something as being a species? And so I'm going to make a little box here. A species is a group of organisms that have the ability to breed and produce fertile offspring. So this is the key. So a group of organisms that have the ability to breed and produce fertile offspring. So if you think about it, so if you think of a mule, for example, cross between a donkey and a horse. Now, a donkey and a horse are not the same species. Yes, they can breed, but they don't produce fertile offspring. And so that's why they're not within the same species. They are separate species because they do not produce fertile offspring. Now, while this is the general definition of a species, you can imagine that this doesn't work in all cases. If we think of bacteria, for example, bacteria reproduce what we call asexually, without a partner. They simply make a copy of themselves and divide. Well, then how do we classify bacteria as species if they don't have the ability to breed and produce fertile offspring? And so other types of things have to go into play in order to classify those species. But as a general rule, generally how we define a species or how we classify something as being in the same species is that these would be a group of organisms that have the ability to breed and produce fertile offspring. Now, does that mean that if, let's say, you're a person and you are infertile, you don't have the ability to have kids, does that mean you're not part of our species? No, right? This is talking about as a whole, the groups as a whole. Do they have the ability to breed and produce fertile offspring? If the answer is yes, they are within the same species. So let's talk about how many species are there. So there are currently approximately 1.8 million different species described. However, estimates suggest that this could be anywhere from there could be 10 million to possibly a billion. There is probably a lot of different organisms that still need to be discovered, and also our understanding of what exactly is a species will also frame how many species there are. If we look at the animals, there are 1.25 million different animal species that have been described. Again, that number is lower than what it probably is. Estimates range anywhere from 10 to 30 million animals potentially. But of the 1.25 million animals that have been described, of that 1.25 million, 900,000 of those are insects. Just think about that for a minute. 900,000 of those animals are insects. And within insects, of those 900,000, a third, or approximately 300,000, are beetles. There are 300,000 different species of beetles. That is mind-blowing if you think about that. If we look at our plants, plants have about 290,000 species that are currently described. If we look at our fungi, there are about 75,000 different fungi. But again, is it more on the upwards of 1.5 million? We're still discovering a lot of different new species. If we look at bacteria, estimates suggest that there might be 2 million in the ocean, maybe 4 million in the soil. There are about 10,000 species of bacteria in a single gram of soil. So if you were to take a single gram of soil, you would find about 10,000 different species. If you look at your hands, the average hand harbors about 150 species of bacteria. And interestingly, when they compared bacteria on left and right hands, the left and the right hand only share about 17% of the same bacteria. So think about that. There's a big difference in the types of bacteria between your left and your right hand. And women tend to have more bacterial types than men. And so there's a lot of diversity. Even in the deep ocean, there can be 20,000 distinct species in 1 liter of seawater. So again, there's a lot of diversity. If you look at people within the class, right? And if you look at people around us, there is a great amount of diversity even within a single species. So why do we have all this diversity? How can we explain all this diversity around us? Well, the tremendous amount of diversity and all of these organisms are basically doing the same thing, obtaining energy to grow and reproduce. But how has so much variation come about and persisted over time? The answer is evolution. There's a tremendous amount of diversity around us because organisms have evolved. evolution over time has changed the way that organisms exist. And so new species have come about as a result of evolution. And so what is evolution? This is going to be what you're going to learn throughout the semester. If we were building a house, evolution would be the mortar. It's not just important to biology, it's central to it. It's one of the main principles that drives biology. And so you guys will learn throughout the semester what is evolution and why does it play such an important role. But evolution is what has driven the change or the number of different species that we've seen. And so one of the things that you need to think about, though, is that if you just simply look at an organism, you can't use physical characteristics to define a species. Think of humans. Humans, there's a lot of diversity. We look very different from one another, yet we are the same species. There are some birds, for example. They're two different species, but they look a lot alike, but they're different species because they don't have the ability to reproduce and produce fertile offspring. So simply looking at organisms is not enough to help us classify organisms into what species they belong to. And throughout the class, you will learn more about how do we classify organisms. So I just want to walk you through an example to kind of talk about classification in terms of humans, right? So domain, remember that there's a 3-domain system. 3 domains, right? The total domains would be bacteria, Archaea, Eukarya. So think for a minute, what domain would we fall into? Answer is, we are domain Eukarya. We are made of eukaryotic cells. Next, we have to think about kingdoms. In domain Eukarya, right, there are 4 main kingdoms, although one is kind of being teased out at the moment. But we have plants, animals, fungi, protists. If you think about us, what would our classification be? We would be Animalia, animals. Phylum, Chordata, the chordates. We have a notochord, and in fact, we also have a vertebrae. So we are within the phylum Chordata. In terms of class, if you look back at the slide I had for classification, you might be able to figure out what class we belong to. So if you need to, take a minute and look back and see if you can figure out what class we are part of. If you look back, you might see we are part of Mammalia. We're mammals. We feed our young with our mammary glands. Order, we have primate, right? And so if you think of primates, you can think of, chimpanzees, lemurs, think of the monkeys and the apes, etc., would be in primates. We are also related. If we look at our DNA sequence, we share about 99% of our DNA with the chimpanzee. 99% of your DNA is shared with a chimpanzee. So we are in the order of primate. Family, Hominidae. So now we're getting more specific into human-like primates. Genus would be Homo, right? And if you have learned about this through our history, you might think of, different types of homos that you've heard of. Homo neanderthal, Homo erectus, Homo sapiens. What are we? What is our species? We are Homo sapiens. Okay, so our species is sapiens. And again, our binomial name is Homo sapiens. Homo is the genus. We capitalize the genus. So notice that this is capitalized. This is the species. Species name is underlined. So Homo sapiens is our binomial name. Homo is our genus. Sapiens is our species. So before we can start to break down and see what makes cell types differently, let's start by talking about things that all cells have. Meaning, if we call something a cell, what structures or molecules will they have that make them be a cell? So there are 4 main things that all cells have. If you would like, take a minute and go ahead and pause the video. See if you can come up with some ideas of things that all cells have. So pause when you're ready, push play, and then listen to the answer. So all cells have, so number 1, chromosomes. And so these are DNA molecules that carry the genes. And the genes basically are going to be the blueprint of the cell. It's how our cell stores genetic information, and that gets passed from one generation to the next. So first thing is going to be our chromosomes. Next, do all cells have a nucleus? Nope. No, not all cells have a nucleus. Remember, we talked about prokaryotic cells do not have a nucleus, so we can't say all cells have a nucleus. Do all cells have a cell wall? Answer is no. All cells do not have a cell wall. Animal cells lack cell walls. Protists can lack cell walls. So we can't say cell walls, but we can say cell membranes. And so the cell membrane is a phospholipid bilayer. It's a type of molecule that separates the living cell from the non-living environment. So cell membranes, a phospholipid bilayer that separates the living cell from the non-living environment. You can think of this as the edge of life. So we have chromosomes. They all have DNA molecules that carry the genes. They have cell membranes. This is a phospholipid bilayer that separates the living cell from the non-living environment. The next one is something that if you've taken a biology class before, you might know this one. And this is a structure used by the cell to make proteins. So what structure do cells use to make proteins? And the answer is ribosomes. So ribosomes are used for protein synthesis. So to make proteins. All cells need to be able to make proteins because proteins are the molecules, the macromolecules, that carry out the functions within the cell. So all cells must have ribosomes because it's used for protein synthesis to make proteins. And the last one is what's called cytosol. And this is the semi-fluid substance inside the cell membrane. So this is our semi-fluid substance inside the cell membrane. You'll hear of the space is referred to as the cytoplasm. The fluid itself is called the cytosol. So technically you could probably say all cells have cytosol or all cells have cytoplasm. Both would be true. But all cells have this semi-fluid substance inside the cell membrane, and that's important to the way that cells work. And so these are things that all cells have. So now what we're going to do is we're going to go through and tease out and figure out what makes cells different, meaning how do we classify something as being domain bacteria versus domain archaea? What are the differences between those cells that allow us to classify them that way? And so that's what we're going to look into. So domain bacteria. They are prokaryotic. So again, they have no nucleus. DNA is naked. It's not within a membrane-bound nucleus. DNA is going to be circular. So they have usually one circular chromosome. You're going to see in a little bit that this is in contrast to our cells, right? Our cells don't have circular DNA. Our cells have linear DNA. They are long strands of DNA. And in our cells, each of our cells has 46 chromosomes, 46 distinct DNA molecules. Bacteria, on the other hand, have circular DNA and usually just one circular chromosome. Some can have more than one, but usually it's one. In terms of cell walls, domain bacteria do have cell walls. Their cell walls contain something called peptidoglycan. One of the things that you'll learn about in biology is to look at the root of the word. Once you start to look at the root of the word and learn what that means, it's much easier to figure out what that means. So when we see peptido, we think protein. Glycan refers to sugar. So what that tells us is that bacteria have a cell wall that is a mixture of sugar and protein. This is going to be distinct to when we learn about organisms in domain Eukarya. The eukaryotic cell walls, if they have them, are usually what we call polysaccharides. They're many sugars linked together. They're not this protein sugar mix. They're simply repeating sugars. But in bacteria, it's peptidoglycan. It's a mix of protein and sugar put together. Now notice I put usually, and the reason I had to put usually is because in biology, in science in general, we can have a rule, but there will always be an exception to that rule. So while we say that bacteria have a cell wall with peptidoglycan, most bacteria do. However, there are exceptions. There's a type of bacteria called mycoplasma. It doesn't even have a cell wall. It's a small bacteria that lacks a cell wall. So that's why it says usually. There are always exceptions to a rule. In terms of bacteria, they are unicellular. If you think of unicycle, what does unicycle refer to? It's one wheel. Unicellular means it's one cell. Unicellular means one cell. So each cell is its own organism. And as a result, bacteria also reproduce asexually, means without a partner. They simply will just make a copy and divide. In terms of how bacteria get their food, some bacteria are photosynthetic, which means that they make their own food. We call organisms that are photosynthetic, we call them autotrophs. Auto means self, troph is feeder. They make their own food. So cyanobacteria that I mentioned is an autotroph. It's able to do photosynthesis and it's able to make its own food. Other bacteria, and probably most bacteria, are what we call heterotrophic. Hetero means other, trophic is referring to feeders. Heterotrophic, other feeders. They have to consume their food in order to survive. So bacteria can be either though. They can be autotrophs or they could be heterotrophs. If we look at structures that are in bacteria, Bacteria are more simplistic than, let's say, a eukaryotic cell. They have a cell membrane or a plasma membrane, and external to that, they have a cell wall. Again, that cell wall is typically going to be a peptidoglycan. They have one circular chromosome. It's not within a membrane. It's in a nucleoid region, so it's in this region of the cell, but it's not separated by a membrane. Bacteria have ribosomes, because remember that all cells have ribosomes in order to synthesize proteins. And then bacteria can have a whole host of optional organelles, meaning that not all bacteria will have these organelles. So we can have a structure that's called fimbriae. E. coli, which is a bacteria in your gut, have fimbriae. It's these little hair-like extensions all over the cell. And the purpose of fimbriae is for attachment. It allows E. coli to attach to the intestinal wall so that it can live in the gut. Some bacteria will produce what's called a capsule. And a capsule is a slime or a capsule. It's a sugary substance that makes the bacteria sticky. And it gives bacteria an advantage in that bacteria are much more resistant to antibiotics, about 1,000 times more resistant if they have a capsule. So it's a structure that gives bacteria an advantage, but not all bacteria have them. A common example that you might know about in terms of bacteria that produce this glycocalyx, the slime on them, would be if you've ever seen a pink slime that is found in the shower. If you've ever seen this in your shower, this pink slime, that's a type of bacteria called Serratia marcescens. And Serratia marcescens produces this slime, and it allows it to stick to the surface of the shower. And Serratia produces that pink pigment, and that's why you see this pink slime. If you've ever gotten this in your bath or your shower, you probably know that it's fairly difficult to get rid of. You might think you clean it, and then it comes right back again. And that's because when you clean that slime layer, you might be getting the outside layers, but you might not penetrate all the way into the inner part of the slime. And so once you get this, it's much more difficult to get rid of. And that's because the bacteria have an advantage by producing this slime. Some bacteria have a structure called flagella. Flagella are these whip-like tail extensions, and they're used for motility or movement. So here are some pictures showing you some diversity within domain bacteria. We have our rod-shaped bacteria like E. coli that's found in your gut. We can have spiral bacteria, what's called a spirochete bacterium. An example, not this one, but another example that you may have heard of before is if you've heard of the sexually transmitted disease syphilis. Syphilis is caused by a bacterial infection. It's caused by a type of organism called Treponema pallidum. That's the name of the bacteria that causes the infection. And you'll notice that if you look along the spiral, you'll see on this diagram it has something called an axial filament. That's basically a flagella that wraps around the bacteria. And the interesting thing about that is that by having that axial filament, think of like if you've ever opened a bottle of wine or you've seen somebody open a bottle of wine and you see a corkscrew, right? And it burrows and it screws its way through to pull the cork out. This bacteria, the Treponema pallidum, the reason that it causes syphilis and it causes the infection is that the spiral bacteria, if you come in contact to somebody else who has this bacteria, so like genital contact, for example, the person who has it has the bacteria on them, and the person who's making contact, when they come in contact, the bacteria is going to transfer from one person to the other, and that corkscrew is going to screw its way, it's going to burrow its way through the skin, and it causes this canker or the sore where the bacteria has entered the body. And so if you screw, you get screwed. So next, the yellow picture, those are bacterial spores. Spores are a structure that bacteria can produce. Again, not all bacteria, but some bacteria. And bacteria produce these in response to a harsh condition. So when their environment becomes unfavorable, they will produce these spores, and these spores are used to help protect them. They're made of a tough keratin protein, just like our skin is made of a tough keratin protein. And so those spores can allow bacteria to survive for millions of years. Think of it as bacteria's way to go into hibernation. It's way to wait it out until conditions become favorable. We have curved rod bacteria, so what we call Vibrio, that's the name of that shape. We have some bacteria that are filamentous. They produce these filaments. These bottom left pictures here, these are two fluorescent bacteria. So these plates were produced by our technician, Dwi Pham, and he basically took this bacteria and he plated it, meaning he used a loop or a needle, so an instrument, to be able to draw on the agar. And this bacteria, this Pseudomonas fluorescens, when you subject it to certain wavelengths of light, it fluoresces. In the ocean, it produces what we call bioluminescence. It causes the bacteria to glow. And so you can see that bacteria can, in fact, glow. Bacteria can produce various pigments. So you'll notice on this bottom right picture, you can see various types of pigments. You can see some that produce yellow pigments. So Staphylococcus aureus, AU, comes from the atomic symbol for gold. Staphylococcus aureus is a skin bacteria, produces a gold pigment. Also on your skin is another yellow type bacteria. It's called Micrococcus luteus. We can have, so in this right one here, this is E. coli. That's bacteria that's found in your gut. We can have this middle picture here, Pseudomonas aeruginosa. Pseudomonas aeruginosa produces this blue pigment. It's kind of this blue-green color. And the reason that Pseudomonas aeruginosa is interesting is it's the most common cause of death in burn victims. It likes water. So if the patient gets a burn and it's wet and it's moist and that bacteria gets in there and it gets trapped in the dressing, it's going to grow in that wound and it's going to cause an infection. Pseudomonas aeruginosa is notoriously difficult to treat with antibiotics. Many antibiotics are not effective. So, and in fact, Pseudomonas aeruginosa can actually, in some instances, grow in Lysol. So in the news, you've been hearing about, Lysol being effective against the coronavirus, right? But in fact, in the case of Pseudomonas aeruginosa, there have been instances where Pseudomonas aeruginosa can actually grow in that Lysol. So it just goes to show you that different chemicals will target different types of organisms. The other thing that's interesting about Pseudomonas aeruginosa is that blue pigment. When that is in the wound, it can cause the wound to have what looks like blue pus. And that's because that pigment from the bacteria, when it interacts with hemoglobin, it gives off this kind of blue appearance. And so it looks like the patient has blue pus. This bottom right, this pink one, Serratia marcescens, right? The one that causes the pink slime in your shower. And so different types of bacteria produce different pigments as well. So next we come to domain Archaea. Domain Archaea, these are prokaryotic. Okay, again, they do not have a nucleus. They have a cell wall. Their cell wall lacks peptidoglycan. It's something structurally similar to peptidoglycan. It's something called pseudomurin. So it's still a protein sugar mix, but it's not peptidoglycan. That's one of the things that makes it distinct from bacteria. It is unicellular, so it does exist as one single cell. It reproduces by binary fission like bacteria do. Again, this is asexual. These organisms, again, tend to be extremophiles. Extreme phile means lover, extreme lovers. They love extreme environments. What do you think a thermophile might like? heat. They grow in extremely hot environments. We have what are called halophiles. They love salt. There's a type of archaea called halobacterium. It's not actually a bacteria, it's an archaea. And it lives on salt crystals. You can find this in Mono Lake, which is in California. And if you go to this lake, you can find these salt crystals that have this pink hue on the salt. That is, in fact, archaea growing on that salt crystal. Most other organisms cannot tolerate high salt environments, and you'll learn why as we go throughout the class. But archaea do have adaptations that allow them to grow in high salt environments in some cases. We have what are called methanogens. These are our methane producers. So this is just kind of an overview of domain archaea. Now, we can't have you classify something as being bacteria or archaea because simply looking at pictures of them is not going to tell you which organism they are. So obviously, we can't ask you to classify domain bacteria and domain archaea. There's no way that you could just look at it and find shared characteristics. So that's why I'm giving you this information myself. Now, when we get into domain Eukarya. Domain Eukarya, again, the cells are made up of eukaryotic cells. So they have linear DNA. So again, long strands of DNA that are found within a nucleus. They're within a membrane. Most cells in domain Eukarya also have membrane-bound organelles. In domain Eukarya, you might have single-celled organisms like amoeba or yeast, or you might have multi-celled organisms like animals, plants, molds, mushrooms, etc. In terms of how they get their food, some are photosynthetic or again autotrophic. They make their own food, while others are heterotrophic. They have to consume their food. Within domain Eukarya, some have cell walls, while others do not. If they do have a cell wall, usually that cell wall, again, is made of a polysaccharide. It's many sugars linked together. So in the case of plants, for example, plants will use cellulose. Or chitin is found in fungi. So if we compare prokaryotic and eukaryotic cells, prokaryotic cells, they have their DNA in a nucleoid region. It's not within a membrane-bound organelle. Eukaryotic cells, DNA is going to be contained within a membrane-bound nucleus. In terms of size, prokaryotic cells are typically much, much smaller relative to eukaryotic cells. Prokaryotic cells, so again, bacteria, archaea, organization, always single-celled. They are unicellular. Eukaryotic cells, they're often multicellular, but they can be unicellular as well. So there's a little more diversity there. In terms of organelles, prokaryotic cells have really only one type of organelle. There are other structures that are like organelles, but not all bacteria necessarily have them. So in prokaryotic cells, they have ribosomes. Eukaryotic cells have many types of organelles. And so if you look back at the previous slide, you can see there's a picture of a plant cell and an animal cell, and there are lots of different membrane-bound organelles that you'll learn about later in the class. So we're done talking about domain bacteria, domain archaea. Now we're going to move into domain Eukarya. So everything else we're going to discuss from this point on is going to fall within domain Eukarya. Now we need to get into kingdoms. So again, you would not be able to classify a protist just by looking at it. So we're not going to ask you to do that, but I still wanted to share some examples of different protists and what they look like. Again, remember that this has now been separated into multiple kingdoms, but for the sake of this class, we'll keep it simple. We'll just put them all in Kingdom Protista for now. We have amoeba. Amoeba are single-celled protists. We have dinoflagellates. We have a paramecium here. We have plasmodium. So this one down here, you see these little ring structures. That is an organism called Plasmodium. Plasmodium falciparum, for example, Plasmodium malariae causes malaria. So you get this disease by getting bit by a female Anopheles mosquito. And when the mosquito bites, she transmits this organism into the body. And eventually it ends up infecting the red blood cells. And it causes this stage that's called a ring stage. It looks like a little ring, and it's infecting those red blood cells, and ultimately it's going to cause malaria. If we look at our algae, red algae, for example, is placed into Kingdom Protista. If we look over here at this bottom right picture, these little purple squigglies, those are an organism called Trypanosoma. Notice that they're in with red blood cells. The interesting thing about trypanosoma in this case causes African sleeping sickness. African sleeping sickness is transmitted by a type of biting fly that's called a tsetse fly. And so you can guess where tsetse flies are found. They're found in Africa. And so these tsetse flies will bite, and the organism will transmit the tsetse fly will transmit the organism into the blood. Now, most organisms cannot survive in the blood because if you think about it, that's where your immune cells are. Your immune cells are your white blood cells. They're in the blood. So normally an organism in the blood is going to be bombarded by white blood cells, and the white blood cells are going to neutralize it and get it out of the body. However, Trypanosoma has a unique adaptation, and that is that it does something called antigenic switches. It changes its outside structure. So the way that our immune system works is that we produce antibodies, and these antibody proteins bind to some surface molecule on the foreign invader. So it recognizes something foreign on Trypanosoma. And it takes time for your body to recognize that and to start making antibodies. By the time your body makes antibodies to neutralize that organism, Trypanosoma does these antigenic switches. It's like it takes off its coat. It takes off its disguise. Now it has a new disguise. It has new antigens on its surface. Immune system starts to recognize it, starts to produce antibodies. By the time it produces antibodies, takes off its coat, it's got another set of antigens on it. So by changing its surface antigens, by changing the molecules on its surface, it's able to evade the immune system. And so when you have Trypanosoma, it ends up being fatal because your body cannot get rid of it. And the reason it's called African sleeping sickness is because this organism stays in your bloodstream and it utilizes your food. If you think about how your cells get the energy that they need, we eat our food, right? It goes through our digestive tract. The molecules get made smaller. Food is absorbed from the digestive tract into the blood. It's going to circulate through the blood to get glucose to all the cells in your body. This organism lives in your bloodstream, though. So what it does is it basically hijacks your food. It hijacks your food that you've just digested, and it uses that glucose for energy. And what ends up happening is that your cells don't get that energy anymore. They don't get that glucose. And so it's called African sleeping sickness because eventually without getting, that glucose, your cells can't make ATP. And these patients will basically slip into a coma. They'll go to sleep and they'll never wake up because their brain is not getting enough food to make ATP. And without making ATP, it doesn't have energy. And that's why patients die from this disease. There are two different strains of the disease. One is more prevalent in West Africa. One is more prevalent in East Africa. one of the strains kills within a couple months, the other one within a couple years. But without treatment, it's always fatal. So within Kingdom Protista, we also have our brown algae and our diatoms. And the reason that they have this brown pigment, so if you look at this bottom picture here, this is what we typically in California call seaweed. It's the stuff that washes up on our beaches. And that's brown algae. And the pigment comes from this chlorophyll A and C and fuchsanthin. So it's a type of pigment that these organisms produce. And as a result, they have this brown appearance that they look like. I will not ask you what pigments they have. I just wanted to point that out, that different color algae use different color pigments, and that's why they appear different colors. If we look at our red algae, for example, they have chlorophyll A and D, and they have an accessory pigment called phycoerythrin, which is what gives it that red color. Green algae historically had been classified with the protists, meaning it was with the other algae, it was with the brown algae and the red algae, but green algae have now been reclassified and have now been classified as being plants. Scientists have decided that they have more in common with land plants than they do the other algae. And in fact, green algae was the precursor to plants, meaning algae evolved and gave rise to the first terrestrial plant. So that is going to be our green algae. And so green algae have chlorophylls A and B, and they have a cell wall made of cellulose. So now I want to walk you through how to use a dichotomous key, because this is what you're going to use, not this exact one, but you're going to use something similar to this to try and help and classify your organisms. So when we talk about a dichotomous key, di means two. There are two choices. And you're going to look at your organisms, and then you're going to figure out, starting at the top, which one of those fits the description. So let's go with this one right here. So start from the top. Pointed ears or rounded ears. So does this one on the left have pointed ears or rounded ears? Pointed ears, right? Notice the ears come to a point. So it says go to number 2. This is number 2. Now we go yellow fur or not yellow fur. Well, it has yellow fur. Therefore, we can see that it's classified. This is rabbit. Let's go to this one. Not going to say its name because I want you to classify it. So starting at the top, round ears or pointed ears. So if we look at the ears are going to be rounded. So it says go to number 4. So you don't go to number 2 this time. You skip all the way down to number 4. 4 says has stripes or does not have stripes. Notice it has stripes. So that's Tigger, right? And so this is how a dichotomous key is. You start from the top, you figure out of those two choices, which one is the best applied to that, and then follow that to the next number. And you keep following it until you get to your classification, or in this case, the name of the Winnie the Pooh character. This is how we use the dichotomous key. So what you're going to do in this is each slide in the PowerPoint has photographs of organisms that belong to a taxonomic group based on their shared characteristics, meaning that they're classified by some particular characteristic. There are a total of 14 different groups for you to identify using the dichotomous key in your lab manual. And in your lab manual, the dichotomous key can be found on page 5. All 14 groups belong to domain Eukarya, right? Because again, I said you're not going to be able to classify things that are bacteria and Archaea. There's just no way. You're not going to be able to look at them to see what shared characteristics they have. So we have not used any of those. They will all fall under domain Eukarya. A hint, there are 4 different groups within Kingdom Plantae. So there are 4 groups of plants, 1 group that belongs to Kingdom Fungi, and 9 different groups within Kingdom Animalia. The groups are randomly numbered, meaning they're mixed up, so they're not in order of the first 4 plants, nothing like that. They're all randomized. And so for each group, you are going to record the group number, the taxonomic name, list a few common names of the organisms, and list at least 2 characteristics that the organisms share in that group. And I will go through and give you an example and walk you through this. So here are some helpful images to kind of help you when using the dichotomous key, because some of these terms might be new for you. If we talk about asymmetry, it means that there's no symmetry, no way that you can divide it into equal parts. If we look at humans, we have what's called bilateral symmetry, right? We can divide us in half long ways. We have a left side and a right side. That's bilateral symmetry. Some organisms have what's called a radial symmetry. Think of it like a pizza. It's round and you can cut it in multiple different directions to make equal halves. So that's symmetry versus asymmetry. Asymmetry means without symmetry. When we talk about worms, some worms are going to be segmented. And what that means is that they have these repeating segments. It almost looks like there's bands, and that's essentially what they are. Those segments are actually repeating subunits that contain, a particular organ, for example, etc. So some worms are going to be segmented. Other worms are what we call unsegmented. It doesn't have those repeating subunits. The other thing that's helpful is when it's asking for like stinging cells. A bee, for example, is not stinging cells. It's a stinger or a sharp structure that's made of many cells. Whereas if we look at the tentacles here, the tentacles of this have each stinging cell is 1 cell. So it has these cells, these nematocysts, that are able to sting. So when we're talking about stinging cells, nematocysts, we're talking about individual cells. So don't think of a honeybee. That's not a cell that stings. It's a structure that stings, but it's not a cell. Jellyfish, for example, have stinging cells. So let's practice with another dichotomous key using actually animals this time. So look at the pictures of these 3 animals. So we're going to start from the top. Has no limbs, so has fins, go to number 2, or has 4 limbs, it's tetrapoda, go to number 3. So which of those characteristics apply? Well, has 4 limbs, right? Each of those has 4 limbs. These are tetrapods. Tetra means 4. They have 4 limbs. So we're going to skip down to number 3. 3 says breathes with gills as a juvenile. So does it breathe with gills as a juvenile, or breathes with lungs as a juvenile? These do not breathe with gills. They breathe with lungs. So we go to number 4. Number 4, skin covered with scales, skin covered with fur. Which of those statements applies to these organisms? Well, they're covered in fur. They're mammalia. So our answer, or taxonomy, would be tetrapoda and mammalia, right? Notice it's tetrapoda and then the class mammalia. Common names. Those are the common names. What do we call them in everyday lingo? So the top one, a cheetah. There's a scientific name for that. We're not asking you for the scientific name. Talking about common names. So we have a cheetah, we have a sea otter, This bottom one is an eastern gray kangaroo. So these are the common names. So when you look at the pictures, the common names are, what do you commonly call these? Shared characteristics. So what would you write in your shared characteristics? Well, has 4 limbs. Notice they've been italicized. Breathes with lungs. And the main one, right, the one that classified them as being a mammal, covered with fur. So these would be the things that you would fill in your table that starts on page 7. So you're going to write in the taxonomy, how they're classified, the common names of some examples, as well as the shared characteristic. And again, you need at least 2 shared characteristics for each one. So we will do this together. So what you want to do is, if you look at table 1-1 on page 7, domain bacteria, domain archaea, those are done for you. Okay, because again, you're not going to be able to classify those. So after archaea, you're going to number your boxes. So the next box down, you're going to put the number 1, and then 2, 3, 4, all the way through 14. You should have 14 total. So we're going to start with number 1. So this is group 1. So these are the organisms on the slide. So this is a practice one that I'm going to do with you. So what you're looking at is we have Ascaris lumbricoides. So this is, if you can tell, they're roundworms. This is actually the most common helminth infection in the world. 1 billion people on the planet are infected with Ascaris lumbricoides. 1 billion. Think about that. 1/7 of the world population has Ascaris lumbricoides in their intestines. It is a very common worm. How do you get Ascaris, or ascariasis is the name of the disease? Well, you get it by eating undercooked food or drinking contaminated water. Well, how do you end up with contaminated food? Well, in some parts of the world, human feces is used as fertilizer because it's rich in nutrients. So if somebody is infected with Ascaris, if they have that worm in their intestine, in their feces are going to be Ascaris eggs. If that feces is used as fertilizer and it's used on the crops like fruits, etc., fruits or vegetables, and let's say that fruit is not washed well or it's not cooked, right? If you eat things that are raw, well, if that fruit or that vegetable is not washed well and you now ingest that vegetable or fruit that's contaminated with the eggs, well, now you're going to end up eventually with Ascaris, these worms, in your intestines. The really gross thing about Ascaris to me is that when you ingest the eggs are going to make their way through the digestive tract. So you're going to start to get these juvenile worms. And the juvenile worms, the interesting thing about them is that the juvenile worms form in the intestine, but they actually burrow out of the intestinal tract into the peritoneal cavity, and they make their way to the lungs. They have a tropism or an affinity towards the lungs. And so those juvenile worms start to develop in the lungs. Now, what can happen, what can make this deadly, is that you can get Ascaris pneumonia, and usually it's not the worms itself that are fatal. Usually it's a secondary infection that occurs as a result of having this worm. So when you get those juvenile worms in the lungs, you can also get a bacterial infection in the lungs. But the gross thing to me is the way that they finish their life cycle is that they will make their way out of the lungs and they will go up the bronchial tree, up, and they will get to your esophagus and you will swallow them and they will finish their development in the intestines. So they're able to crawl their way out of the lungs, up the neck, and down the esophagus and into your stomach. And then they develop into the adults in the intestine, and then the eggs get passed in feces again. That is the most common helminth infection in the world. In the US, the most common helminth infection, pinworm. Pinworm, they're little tiny worms. They're about the size of a grain of rice. Pinworms are most likely or more often transmitted by children. And the reason for that, and again, this is gross, so sorry if you haven't eaten yet, but the way that a pinworm infection gets perpetuated is that if somebody has pinworms in their intestine, these pinworms reside in the intestine. At night, the females crawl out of the intestine, out the rectum, out the anus, and she likes to lay her eggs on the perianal region. That's the region between your anus and your private parts. So that little taint, that skin there, is where the female likes to lay her eggs. So she crawls out at night, she lays her eggs on the perianal region, she goes back up and into the intestine. Now, one of the main symptoms of pinworm, itchy rectum. It makes the rectum itch. Kids are especially bad about this. Their butt itches, they're going to itch it. So what ends up happening is the kid wakes up in the morning and their rectum itches and they scratch. Now they have eggs all over their hands. They touch surfaces around the house. It's in their bedding. It's in their towels. These eggs are all over. If you come along and you touch those eggs or you happen to inhale those eggs, let's say you're, changing the bedding and those eggs go flying and you inhale those eggs, those eggs, when you ingest them, will form those pinworms in your intestine. And so that is why children are more likely to transmit them, because kids are more likely to not understand that you shouldn't, touch your butt and then touch other surfaces. Most adults know that that's not sanitary. Kids don't really understand. Something itches, they itch. They go around, they touch different things, and pinworm gets perpetuated that way. So it's often transmitted by children, so in a daycare facility or an orphanage or something along those lines. Not always, but most common. And pinworm is the most common worm infection in the US. Not in the world. Ascaris is the one in the world. Pinworm is in the US. We have C. elegans. C. elegans is a type of worm that's studied for development because we have mapped the different cells and where they formed from. We have a worm that causes elephantiasis. Notice that it blocks these lymph nodes and you get swelling in these limbs. So let's start by practicing. Now that we've looked at these worms, let's go ahead and go through the classification. So we're starting with the Dichotomous Key from the lab manual. So start at the top, number 1. Organisms are prokaryotic. Most are single-celled and lack a nucleus. Or organisms are eukaryotic, single or multi-celled and contain a nucleus. Now remember I said all organisms in this are in domain Eukarya. So you're going to write under classification, domain Eukarya. Go to number 3. So we go to number 3. There we go. Go to number 3. Number 3 says cells contain chlorophyll A and B. So those green pigments within a chloroplast. If that's true, they're going to be in kingdom Plantae. So are these plants? Or cells do not contain chlorophyll. Go to number 7. Are these plants? Answer is no. Go to number 7. So we skip 4 through 6. We're going all the way to number 7. Number 7, I accidentally put on there already. Organisms have cells surrounded by a cell membrane and a cell wall, right? So if the cell is rather rigid, it would have a cell wall. It would be kingdom fungi. Or 7b, organisms have cells lacking a cell wall. Organisms can move. They don't have to move, but they can in most cases. Kingdom Animalia, go to number 8. So these are flexible, right? They can move. So under classification, Kingdom Animalia, go to number 8. 8 says, organisms are covered with pores and lack symmetry. They're asymmetrical. Remember what that means is that you can't divide them in half in any way. Or 8B, organisms are not covered with pores and the body is symmetrical. Answer is body is symmetrical, right? They have bilateral symmetry. They can be divided in half. So we have symmetrical. Go to number 9. Organisms contain stinging cells. So again, not a stinging organ, but stinging cells, nematocysts. Or organisms lack stinging cells. So these worms, they don't have stinging cells, so they lack stinging cells. Go to number 10. All of the organisms lack a hard skeleton and are worm-like. Go to number 11. Or most of the organisms in the group have a hard skeleton, either internal or external. Go to number 13. These are worm-like. Go to number 11. 11. Organisms have segmented bodies, so repeating structures. So again, you would see those lines through it. So the repeating structures. Or organisms lack segments and are smooth. Go to number 12. If we look at these worms, notice they don't have segmented, right? So they don't have segmented bodies, so they're smooth. Go to number 12. Organisms have a flat body. Organisms have a round body and pointed ends. So if we look at these pictures, are they flat or are they round? And you can see that they're pretty round and pointed ends. Phylum Nematoda. So for group number 1, what you're going to write in your table, you're going to write next to number 1, domain Eukarya, kingdom Animalia, phylum Nematoda. Those are all the classifications we got as we went through this process. Common names. So we have roundworms, we have Ascaris, we have Eukarya, we can have, I won't make you know the one for pinworm, but those are just some examples. You could even write pinworm there. Shared characteristic. Multicellular, right? These exist as more than one cell. They are worms. They lack segments, and they have round body with pointed ends. A good rule of thumb, whatever you got that got you to your classification is definitely what you want to have in your shared characteristic, because that is the shared characteristic that helps you to classify that organism. So that definitely needs to be in your shared characteristic. The other one are things along the way that you might have come across that make it unique. So worms, they're not segmented worms, and they're round body with pointed ends. So those would be the things you would put in your shared characteristics. And so this is just an example of how you're going to do this lab. So now for the rest of these, you're going to go through and you're going to look at the organisms on the slide. You're going to use the dichotomous key, and then you're going to write the taxonomic name when you go through your dichotomous key, the common names, so the common names of some of those organisms, and then the shared characteristics. So let me show you. So this was group 1. Group 1 was done for you. You're going to do group 2. So you want to look at these organisms, see what they have in common, write some common names, right? So you can name some of these probably on your own pretty easily. And then using your dichotomous key, what are the shared characteristics? So what I'm going to do is I'm going to go through these fairly quickly in the video. You have 2 options in order to study these. One would be to pause the video and do it while this is paused. The other option is simply to look at the PowerPoint and look at the pictures so that they're not going from one to the next, and work your way through the PowerPoint. But basically the images are going to be the same. So this is group 2. You need to classify this. This is group 3. This is group 4. This is group 5. This is group 6. Group 7. Group 8. Group 9. So these little plants. This is group 10. This is group 11. This is group 12. This is group 13. And this is group 14. So go through the slides. You're going to view these pictures, and then you're going to turn in your lab on the indicated day. And the next class period, we will go over the answers with you before you take your quiz the next time. So you need to finish this before the due date and then submit it. And then we will go over the answers together so that you know how to study these. But you need to go in and fill in this table. And so this is what you're doing for Lab 1. We're classifying organisms based on shared characteristics.